Hi, I am Phoebe Leung from Yinghua Girls School,
Hi, I'm Sydney Lee from St. Paul's Coeducational College.
and our project is uncovering the most efficient setup for microbial electrolysis cells for hydrogen production. Hydrogen is a very promising source of clean energy, but it does not exist in its pure form in nature. To purify it, we either need to do electrolysis, which needs a lot of energy, or fermentation, which is very slow. However, there's recently a new form of hydrogen production that is developed, microbial electrolysis cells, or MECs for short. It combines both methods of fermentation and electrolysis. A small current is added to a fermentation setup to boost speed and to use less energy than electrolysis. Our objective is to improve on MEC technology, which is quite new with a lot of room for R&D. However, due to time constraints, we could not complete the project in its entirety. Instead of testing all the variables that affect the efficiency of an MEC, we changed our plans to designing an MEC with a theoretically most ideal combination of variables. We then compared the results of that against regular electrolysis, or at least that was the plan. However, we couldn't source and purify the bacteria for the MEC in time, and therefore couldn't test our setup. So what we did was the control experiment of the regular electrolysis and design our MEC. And here's our experiment testing the efficiency of electrolysis. This was our setup, and we compared the net energy produced by comparing the energy produced by finding the volume of hydrogen we made, multiplying that by the density of hydrogen, and multiplying that by the amount of energy produced per unit mass of hydrogen. We then subtracted from that the energy consumed during electrolysis right here. However, we found that during all of our different voltages that we tested for electrolysis, the net energy produced we found was negative. However, luckily for us, they were all different. So we could plug the results of our data into a graphing calculator to find the relationship between the voltage used in electrolysis and the net energy produced. And then we use that to find the point at which the net energy produced switches from negative to positive. And I'll now pass the time to my partner to talk about the results.
So as my partner said, the reaction of the electrolysis cell was pretty slow, and the tests on them yielded results which were negative. So for the MEC to be more effective than regular electrolysis, it would need to reach the point where more energy is produced and consumed in 1,580 seconds for 1.5 milliliters of water. So we have figured out some factors affecting the efficiency of MECs. The theoretically most ideal setup has these components as highlighted in yellow: seawater, glucose, one chamber, cyanobacteria, carbon/graphite, and stainless steel. So here's our experimental setup for the theoretically most ideal MEC setup. Regarding procedures, the method of figuring out the efficiency of the cell is the same as the electrolysis cell, but the main difference is that the setup is an MEC setup instead of an electrolysis setup. Summing up our research project, expectation: we expected to test out the theoretically perfect model for the MEC and compare that to regular electrolysis. Reality: that couldn't be done because of time constraints, so we drafted a design for the theoretically most ideal MEC, and we compared various factors that would affect the efficiency of the MEC, and we found the efficiency of regular electrolysis.
And that is our project. It was brought to you by other scientists. Here are our sources. While working on the project, I definitely learned not to be too ambitious and learned that complex projects take time. While working on the project, I learned that it's okay not to complete everything in a very short time frame like this program, because our project was definitely quite long and complex with a lot of procedures, and it was not realistic to expect to complete everything in time.
During our project, we definitely encountered a lot of difficulties and challenges, with the one being not able to source cyanobacteria being the biggest. It was really devastating at first because cyanobacteria was basically the center of the base of our project, but I'm really glad that we didn't give up and we eventually figured out a solution to finish our project, and I'm really grateful for everything, and it's a really memorable experience. That's the end of our presentation. Thank you so much for listening. We hope you enjoyed it.